Even in humans, cognitive mapping is a hotly debated topic. The main point of contention is how mental representations of spatial information are organized. One proposal is that humans form cognitive maps of different environments, but have difficulty combining these, even if they are linked in the real world. Wang and Brockmole, 2003, examined this by asking individuals to imagine navigating between small and large spaces in their environment, e.g. within a room in a particular building, versus that building within a larger complex of buildings. They concluded that individuals switch between spatial representations as they move through an environment, mentally situating themselves within the small or large space, but not both. For example, finding a room within a building, then choosing which door of the building provides the most direct access to another building, involved shifting from one cognitive map to another. According to the authors, this shift is mediated, or at least facilitated, by language, suggesting that the ability may be unique to humans. A complementary line of research focuses on understanding how other cognitive processes, such as memory, relate to spatial mapping. These processes must interact because cognitive maps will be modified and updated as organisms move through their environment. There is little doubt that humans and animals know where they are in relationship to spatial cues in their environment. Whether they do so by referring to a cognitive map is unresolved. Interactive systems. The preceding sections emphasize that most species can use different cues during small-scale navigation, even if they have a tendency to employ one strategy over another. This predisposition to rely on a particular cognitive process often reflects the ecological constraints of a species. For example, food-caching animals may use some form of a cognitive map rather than environmental geometry to locate food because they regularly approach hidden stores from different directions. In addition, exposure to specific cues during development, as well as training in a particular paradigm, can influence which cognitive mechanism animals use during navigation. If animals learn that one set of cues, e.g. landmarks, are not reliable predictors of a goal location, they may revert to an alternative strategy, e.g. environmental geometry, even if they were unlikely to do so in the natural environment. The fact that many species can use spatial information in flexible ways allows them to adapt to changing environmental conditions, some of which involve endogenous physiological signals. See Box 5.2. Box 5.2, Hormonal Modulation of Spatial Navigation. Box 5.1 outlined evidence for sex differences in spatial navigation, more accurately described as a preferential reliance on directional cues in males and positional cues in females. But this distinction is not absolute. In many species, males outperform females on spatial memory tasks only at certain times of the year. This observation led Galea and colleagues to speculate that changes in circulating levels of sex hormones underlie a male superiority on laboratory tests of spatial memory. 1996. To test this idea, they compared male and female performance in the water maze across reproductive cycles. Female deer mice, Paromyscus maniculatus, were worse at finding the hidden platform during the breeding season, whereas males of this species were better. In both males and females, performance was inversely related to circulating levels of the female sex hormone, estradiol. In contrast, higher exposure to testosterone, the primary male sex hormone, in utero, was associated with improved performance. Importantly, sex differences were not apparent prior to puberty, suggesting that differences in spatial memory emerge when sex hormones are activated. An important aspect of Galea's study is that she tested spatial memory under conditions of naturally fluctuating hormones. Manipulations that artificially block or enhance these levels often produce contradictory effects because they push hormones beyond a normal physiological range. Although they were specifically interested in proximate mechanisms of spatial navigation, Galea and colleagues provide an ultimate explanation for their findings that increasing levels of male and female sex hormones have opposite effects on spatial navigation. improving and disrupting this ability respectively. They note that male reproductive behaviors would be facilitated by increased spatial exploration as animals search for mates, but female reproductive strategies would favor staying close to home. This is particularly true when females are close to giving birth, which may explain why women show deficits in spatial memory tasks at the later stages of pregnancy. Indeed, Lisa Gelia often describes her findings in terms of her own experience, desperately searching for her car in the parking lot when she was 9 months pregnant. Once it became clear that animals have different mechanisms of small-scale navigation at their disposal, researchers began to investigate how these function together. One suggestion is that navigation is guided by either landmarks or environmental geometry. Animals may switch between the two as they move through the environment, but only one of the processes is directing behavior at a given point in time. Evidence supporting this claim comes from experiments using the Q competition paradigm described in section 5.3. In general, learning to navigate using landmarks makes it difficult to find a goal using geometric cues and vice versa. As with landmarks versus beacons discussed previously, animals appear to rely on one strategy or the other, but do not combine information from the two, Shuttleworth 2010. It is unlikely that this distinction reflects an inability to use multiple sources of spatial information, because food-caching birds, at least Clark's nutcrackers, are more accurate in retrieving food as the number of landmarks increases. In other words, cognitive load is not a problem in these tasks. It is the cognitive process of using landmarks to locate a goal that operates independently of navigation by environmental geometry. In contrast, path integration and cognitive mapping may work in parallel to landmark or environmental geometry navigation. It makes sense that these processes operate simultaneously, as cognitive maps, if they exist, are a combined representation of landmarks and environmental geometry. Path integration may work in a different way, acting as a backup system to landmark navigation when animals get off track. This implies that computations that would allow animals to return home by a direct route are ongoing, but only come into play if cues in the environment are unreliable or unstable. Even desert ants appear to use landmark cues preferentially, only resorting to path integration if they are displaced from their traveling route. In sum, small-scale navigation is accomplished in a variety of ways. Animals may locate a goal using landmarks, environmental geometry, path integration, or cognitive maps. Although a particular species may exhibit a tendency to rely on one process or another, most animals can navigate using more than one of these mechanisms. Frequently, animals switch between strategies, depending on how close they are to the goal and what they have learned about spatial navigation in the past. When cognitive processes controlling navigation are activated simultaneously, they may compete to control behavior, e.g. landmarks and environmental geometry, interact cooperatively, e.g. landmarks and environmental geometry facilitate cognitive maps, or function in a hierarchy, e.g. landmarks override path integration. These interactions highlight the flexibility of small-scale navigation, in that the ability to rely on different processes allows animals to adjust their behavior to the ever-changing world around them. Large-scale navigation. Large-scale navigation is defined as long-distance travel to a goal, often through unfamiliar territory. Distinguishing large from small-scale navigation is not always easy, as it can be difficult to know whether a particular environment is unfamiliar to animals not reared in captivity. Moreover, the difference between short and long distances will depend on the species under study, e.g. ants versus birds. Nonetheless, small and large-scale navigation are usually examined independently, even if researchers agree that the two may involve overlapping mechanisms. Homing versus migration. One of the amazing feats of large-scale navigation is homing, the ability to return to a nest or burrow after displacement to a distant, often unfamiliar site. Humans exploited the homing abilities of birds during the 19th and early 20th centuries. During the 1800s and early 1900s, by sending messages over long distances attached to the legs of homing pigeons, Columba livia, appropriately named carrier or messenger pigeons. These displaced birds were able to return, more or less directly, to a star location, suggesting that cognitive processes supporting this behavior provided some fitness advantage in the past. One of these relates to breeding, which is expressed as natal homing, returning to one's birthplace to reproduce. Natal homing occurs most commonly in aquatic animals such as green sea turtles, Chelonia mitis. These animals hatch at night on a sandy beach and head directly to the water, where they spend most of their life swimming in open seas and feeding at distant locations, sometimes at the other side of the Pacific Ocean from where they were born. The beaches that are ideal for feeding are not the same as those needed for nesting, so 3 or 4 decades later, when females are ready to reproduce, they return to their natal site to lay eggs. In all likelihood, this long-distance travel between feeding and birthing sites did not arise spontaneously. Although the two locations are now separated by an ocean, they may have been closer together when turtles evolved more than 100 million years ago. Natal homing of Pacific salmon, genus Oncorhynchus, that includes 8 different species, is just as amazing. They spawn in freshwater streams that feed into the ocean, spend 4 or 5 years swimming in the Pacific, then return to their birthplace when they reach sexual maturity. These journeys can be arduous, covering up to a few thousand kilometers, much of it upstream. See Figure 5.11. Figure 5.11, salmon jumping up a waterfall to spawn near Katmai National Park, Alaska. Migration, large-scale seasonal movement between spatially distinct habitats, can be even more physically demanding. Migration of the bar-tailed godwit, Lamosa laponica, includes an 11,000-kilometer non-stop flight from Alaska to New Zealand with no pause for feeding. Humpback whales, Megoptera novaeangelia, feed in cooler northern oceans and travel up to 25,000 kilometers each winter to reach warmer waters where they breed and give birth. The longest known migratory route of a land mammal is undertaken by tundra caribou, Rangifer terendus, in northern Canada, which travel approximately 5,000 kilometers between summer and winter habitats, covering 25 to 50 kilometers per day. Finally, insects such as monarch butterflies, Danaus plexippus, display an amazing transgenerational migration in which a single butterfly partakes in only one segment of the Canada to Mexico journey. The insects reproduce along the way, and successive generations travel to the next migration stop. Despite the obvious similarities, homing and migration do not always occur in the same groups of animals. Some animals that migrate do not exhibit homing, and vice versa. One suggestion is that homing and migration may have evolved from a common process, with differential evolutionary pressures leading to homing in some species and migration in others. Indeed, many researchers believe that homing and migration involve similar navigational mechanisms, particularly in avian species. For this reason, large-scale navigation is discussed in this chapter as one topic, which includes both homing and migration. The next section examines cognitive mechanisms that underlie both behaviors, with an emphasis on similarities and differences between the two. Mechanisms. The cognitive mechanisms mediating large-scale navigation must provide animals with information on when to leave, which direction to go, and where to stop. These processes are frequently studied in birds using an Emlyn funnel. Emlyn and Emlyn, 1966. A small circular bird cage with a funnel-like top that is covered by glass or a wire screen. See Figure 5.12. An ink pad is placed at the bottom of the cage so that foot traces are left on the sloping sides of the cage as birds hop about or try to escape. Using this setup, Gwinner, 1977, showed that the nighttime activity of migratory species reared in captivity increases around their usual time of migration, suggesting that these birds possess an endogenous, i.e. genetic, mechanism that keeps track of yearly cycles. This migratory restlessness, usually referred to by the German term Zugunruhe, is synchronized with external cues marking seasonality, primarily changes in daylight, although many species display the behavior even when these cues are absent in the lab. Migratory onset in non-avian species is mediated by a similar mechanism, an endogenous annual clock that interacts with environmental changes, such as a reduction in the length and intensity of daylight. The initiation of natal homing depends on the same signals, combined with changes in circulating levels of reproductive hormones. Figure 5.12, schematic of an Emlyn funnel, used to assess spatial orientation during migration in birds. Birds are placed in an inverted cone with an ink pad lining the bottom of the cage. When the bird hops or flutters up the sloping walls, traces of ink are left on the funnel sides. The top of the cage is clear so that the bird's image of the night sky, e.g. star patterns, can be manipulated. Oh, that's so cute. So there's an ink pad at the bottom and then there's the paper on the funnel walls to pick up the ink. Okay. As with timing, the direction of migratory travel appears to be under endogenous control. This is manifested in the natural environment when first-time migrants successfully travel to their winter destination on their own. This does not occur in all migratory species, but the fact that it occurs at all is impressive. Helbig, 1991, tested inheritance of migratory direction in two populations of European blackcaps, Sylvia and Trichipala, that migrate to either Spain or Africa. As expected, young birds who had never migrated exhibited zaganra toward the winter destination of their parent population. Crossbreeding the animals resulted in a direction of travel almost exactly midway between the two. Subsequent research, using a variety of techniques, confirmed a genetic contribution to migratory direction in birds, a principle that likely applies to other animals. From a cognitive perspective, the question of interest is how these endogenous signals point animals in the right direction. The answer, on a general level, is that migrating and homing animals are equipped with a global reference system that allows them to determine their own position in relationship to some external referent. These referents can be divided into four separate topics. Sun compass. Kramer, 1952, was the first to suggest that birds, specifically European starlings, Sturnus vulgaris, use the sun's position to maintain a constant direction during migration. This sun compass hypothesis was supported by a series of studies examining flight orientation of captive birds under different solar conditions. The majority of these studies used a phase shift manipulation in which the light-dark cycle in the colony room was out of sync with the natural environment, e.g., rather than daylight beginning at 6 a.m. and ending at 6 p.m., lights in the animal house turn on at 2 p.m. and off at 2 a.m. If this cycle continues for more than a few days, an animal's circadian rhythm will shift, as will a human's. Under these conditions, animals made errors in migration orientation that were predicted by the difference between where the sun was and where the sun should be, given the animal's circadian cycle. See Figure 5.13. Thus, cognitive processing that allows birds to use the sun for navigation relies on an internal clock that keeps track of the sun's position over a 24-hour period. Amazingly, the timing mechanism also takes into account seasonal shifts in the sun's position. Figure 5.13, effects of a phase shift manipulation on sun compass orientation of homing pigeons. A. Under natural conditions, circadian rhythms peak at midday when the sun is in the south. Pigeons use this information during migration to orient in a southerly direction. B. If pigeons are housed in a light-dark cycle that is advanced 6 hours, their circadian rhythm is shifted, so the midday peak now occurs during the natural morning. When released, birds orient toward the sun, which is now in the east. Okay, so you can see the circadian rhythm peak at about 12 noon when the sun is in the south, and so the pigeons orient to the south, and then when this is phase shifted, the circadian rhythm peaks at about 6 a.m., so birds will orient to the sun again, but it is now in the east instead. Insects are also capable of using a time-compensated sun compass in large-scale navigation. For example, monarch butterflies will orient in the wrong direction during migration if they have undergone a phase shift manipulation. Although they do not migrate, ants and honeybees keep track of the sun's position across the day and use this information to return home from foraging trips. Thus, there is at least one common mechanism of small and large-scale navigation in insects. Some food-caching birds appear to use a sun compass to locate hidden stores, although the evidence for this claim is still preliminary. Gold et al. 2010. Similarly, there are relatively few studies examining whether mammals use the sun as a directional cue during navigation. The sun may be less important for navigation in mammalian species because it is not a reliable cue in poor weather conditions or when animals are behind large landmarks, e.g. a mountain. This limitation is overcome in insects that have evolved a sensory system to detect polarized light. Polarized light is produced by the sun's rays hitting the Earth's atmosphere. The resulting three-dimensional pattern varies as the sun moves across the sky, making it a reliable time-of-day cue even when the sun is obscured. Some vertebrates, including certain species of fish and amphibians, appear able to detect polarized light, but, given the biological structure of the eye, it is unlikely that this ability is present in mammals.